Our next speaker is Laura Niedernhofer from the University of Minnesota.
Well, thank you very much, Daniela, and also to Morten. It's absolutely a pleasure to be here in person, and I have to say that I think you've created the best clock I can think of for monitoring the exponential momentum in this field, so thank you. So today, I'm not going to talk about any particular compounds or interventions, but really want to focus on a platform of preclinical models that we've been developing. But I'd like to first start, and then I will end with just one slide to bring up some maybe food for thought or provocative ideas. Okay, that's not working. So I think we have to think beyond chemotherapy-derived accelerated aging. So there's lots and lots of disease survivors who are aging very, very rapidly. So Marco beautifully talked about these premature aging and cancer survivors who were treated as children with genotoxic agents. But let me also bring to your attention that many, many genetic disorders, these patients are surviving decades longer than when I started my career. We also have many, many folks dealing with chronic metabolic diseases, and they're aging more rapidly. And also think about all of our transplant patients. They are all aging rapidly. So my point is, I think we're still underestimating the challenges we are facing in treating accelerated aging, or the big problem of aging biology. But the positive side of this is they do offer other opportunities for clinical interventions and trials to test geroprotectors. So now back to my main point, and this is the basis for our preclinical model platform is we're starting from a vantage point of genotoxic stress or genomic instability. And we all know that drives very potently cellular senescence, and so we use this platform initially to screen for xenotherapeutics, as Paul Robbins described earlier. And the background for all of this is a progeroid syndrome that I helped to discover when I was in Jan Hoijmaker's laboratory in Rotterdam, the Netherlands, for my postdoctoral fellowship. And this is driven by mutations in one of two genes, either XPF or ERCC1. And this progeroid syndrome is characterized by normal early development, but then these children age extremely rapidly. And what's pretty unique about this rare progeroid syndrome is these patients have effects in virtually every organ system that you can think of. So it's quite systemic. So what do ERCC1 and XPF encode? Well, two subunits of a singular DNA repair endonuclease. So these two proteins bind one another, and what's very important is they stabilize one another in vivo. So if you knock out ERCC1, as was done in this case, and you look in tissues of these animals, they don't have XPF either. So we ascribe all of the phenotypes of progeria to loss of the holoenzyme and therefore loss of the DNA repair capacity attached to ERCC1 XPF. So I spent a big chunk of my career trying to figure out what was so special about ERCC1 XPF. And what is special about it is it's involved in at least 3 of 5 major DNA repair pathways. So nucleotide excision repair, cross-link repair, and repair of some double-strand breaks. And another important point I'd like to bring to your attention is when we look for these proteins, we only ever find them in nuclear fractions of cells or tissues. So their sole job is to protect the nuclear genome against genotoxic stress. So again, we're ascribing all the phenotypes associated with this progeria to failure to maintain nuclear genome stability. So while in Jan's lab, I created a mouse model of this human disease. And this is the ERCC1 minus delta mouse model that I'm going to talk about extensively. It's the combination of a knockout and hypomorphic allele of ERCC1. So just like the patients, they express about 5% of the normal complement of this DNA repair endonuclease. Mice are adults at 9 weeks of age, so these animals reach adulthood looking pretty darn good, but then they fall apart quite rapidly. And without touching these mice, spontaneously, they develop virtually every age-related disease that I fear. So this includes cognitive decline, loss of vision and hearing, pulmonary fibrosis, heart failure, hepatic fibrosis, disc degeneration, chronic kidney disease, osteoporosis, loss of subcutaneous fat, urinary incontinence, fatty replacement of the bone marrow, sarcopenia, and peripheral neuropathy. So again, spontaneous phenotypes, and these have been really valuable models then for testing a lot of hypotheses about age-related diseases and how to intervene. So another important point about these animals, we do everything in an F1 background, so they're not inbred weird, and they only have a lifespan about 6 or 7 months, so we think they age about 6 times faster than wild-type mice. So I think 2 experiments that were kind of critical to me believing in these animals was first showing that they do have increased DNA damage in their tissues. So this is work done by Yin-Sheng Wang in collaboration with us. He builds these incredibly robust assays that are LC-MS-based to measure endogenous DNA lesions that are very low in abundance. So these are 4 cyclopurine addicts that he helped us measure in tissues of our animals. They are oxidative DNA lesions that form spontaneously and can be detected in human samples. So when he looked at these in renal tissue of our animals, each of these bars represents the sum total of all 4 of these addicts. You see in the young animals, which are, the mutant ones are in red, that at a young age, when the animals do not have aging symptoms, they actually do not have increased oxidative DNA damage. But when you look in older animals that are frankly progeroid, they do have significantly increased levels of oxidative DNA lesions compared to their wild-type littermates. But what's really important is the bar on the right-hand side of this graph, which shows that wild-type mice, when they get old, have the same amount of these lesions. So even if you're completely DNA repair competent, you will accumulate oxidative DNA damage over time. So what we think is happening in these animals is just a compression of normal physiology. It just happens very quickly. So because DNA damage is such a potent driver of senescence, we began to look in a variety of different tissues of our animals. And across these 13 different tissues listed on the left, we measured P16 messenger RNA, P21, a variety of other markers. And this is just a heat map showing those results. So everything is relative to young wild-type mice, and you see old wild-type in the middle as a reference range. And our progeroid animals, when they're 6 times younger than the old wild-type mice, they basically have the same pattern of senescence. So the heat map is very similar in these progeroid mice, and never does the senescence level truly exceed what happens in normal aging. The same was true if we look at a heat map of P21. Very similar between old natural aging and accelerated aging. And of course, we have done this for a variety of different senescence markers. So again, we think endogenous DNA damage is driving senescence, and this happens with normal aging. We've just got a system where it happens quickly. So when we combine these animals then with a variety of different reporter systems, we really can get answers about, is an intervention slowing senescence or aging quite quickly? So in this case, we used Ned Sharpless P16 reporter mouse with a luciferase. And what the graph is showing is intermittent exposure of these animals to fisetin, a senolytic that Paul and I helped discover with Tamara Ciccone and Jim Kirkland. So we're putting it in a chow, we give it to them intermittently, and you can see in a matter of weeks, we know that this intervention is actually suppressing P16 messenger RNA and thereby senescence. And the effect is held even when the animals are not directly exposed to those drugs. So again, a really quick model for screening drugs. So what I've shown you up to this point is that ERCC1 mice have genomic instability, they have cellular senescence, but what about other markers, hallmarks of aging? And so what I want to do is just show you that these animals actually have a lot of these different markers of or hallmarks of aging, even though we're starting from a vantage point of genome instability. And so I'm kind of going to go clockwise around this wheel very quickly with just a smattering of data. So the first thing we assumed is that telomeres would be affected in these mice. And we paired with Titia DeLonge to help us look very critically at this. And in fact, that is not the case. So their telomeres are perfectly fine, and that's in mice or in humans. So what about epigenetics? So Caroline Soda-Palma in my laboratory has looked at this particular histone mark in tissues and cells of our mice. This is a transcriptional repressor largely. And she found that in primary MEFs from these animals, that there is indeed reduced levels of this histone mark, particularly under situations of oxidative stress. This can be restored if you inhibit a particular lysine demethylase, LSD1, and this is true in either a dose-dependent or time-dependent manner. We found that in animals and animals and cells that are treated with this LSD1 inhibitor, we actually attenuate senescence and SASP. So P16 goes down dramatically, although P21 does not, and a number of SAS factors do as well. What about nutrient sensing and autophagy? So in the top Western blot, what I'm showing you is elevated mTOR levels, phospho-mTOR, in muscle stem cells derived from the minus delta mice. And what's interesting is if you treat these cells in vitro with rapamycin, you actually see a suppression of mTOR signaling. And if we look at autophagy, looking at the ratio of LC3 to LC3-2, then you see, again, a depletion in the minus delta that can be restored by rapamycin. So there's certainly defects in nutrient sensing and autophagy in these animals. Mitochondria are also affected, even though I told you ERCC1-XPF doesn't go anywhere near mitochondria. So we look at cells from these animals and staining those for mitosocs, and the yellow color indicates mitochondria with increased ROS. When we look at with super resolution microscopy, the mitochondria look horrid under situations of oxidative stress in the bottom panel. And even when we look in tissues, say liver of these mice, we find that there's an increased abundance of superoxide anion and also evidence of damage of other macromolecules in the cells, so in this case, lipid peroxidation. And I should point out this all happens with normal aging. That's the blue bar on the right. So again, we can resolve this by treating our animals with a novel mitochondrial targeted radical scavenger. There's the structure of it. It's well published if you're interested. When we treated those P16 luciferase reporter mice with this drug, we found decreased cellular senescence in the animals that was sustainable. And this was even good enough to suppress pathology in these animals. So they had decreased osteoporosis, as indicated by the micro-CT of the vertebra, as well as decreased neurodegeneration, as indicated by GFAP staining of the cerebellum. Stem cells are even altered in these animals. So this was work done by Mitra Lavasani at Northwestern University. She looks particularly at muscle-derived stem progenitor cells. And this is an experiment where we challenge the muscle with a toxin. And what we find is just like with normal aging, there's impaired regeneration. So you see a lot of blue color, which is simply fibrosis instead of healthy muscle coming back. Now, as Paul showed you earlier, if you transplant back muscle-derived stem cell progenitors, you can actually rescue this phenotype. So this was true in lifespan extension of ERCC1 knockout mice, as well as health span extension in the ERCC1 minus delta mice. So their stem cells are certainly not happy. And finally, we see some inflammation in these animals. Nobody's surprised by that. In particular, in this case, we were using an NF-kappa B reporter system. So you see green or GFP expression if NF-kappa B transcription factor is activated. So up top, you see a lot of green in the renal tissue of old wild-type mice, and this is particularly in the glomeruli. And then we also see a lot of green in the right-hand panels of ERCC1 minus delta and knockout animals. And when we quantitated this, and again, a study with Paul Robbins, we see elevated levels of NF-kappa B activation and inflammation in kidney, muscle, pancreas, and liver, much to our surprise, not in the spleen, which is where you'd kind of expect NF-kappa B to be activated in the immune system. Again, this is reversible. So Paul and his postdoctoral fellow Lei Zhang developed a small molecule inhibitor of NF-kappa B activation, and this is adequate to suppress senescence markers like P16, P21, and SASP. And it's also adequate to extend the health span of these ERCC1 minus delta mice. So if they're chronically treated with a small molecule, we get lower symptom scores or extended health span. So what I've shown you is, of all the hallmarks that we've looked at, other than telomeres, many of these are altered in the ERCC1 minus delta mice. And furthermore, treating each of these different hallmarks really does have a positive effect. So I think we need to be thinking in terms of there's lots of ways to attenuate aging biology, even if you're starting with a primary defect that's very specific. So what I wanted to say beyond this is we've got a ton of mice. So if you're interested, please contact me. We've gotten really bold and really pricey. And we've taken these accelerated aging mice, and we're now adding all sorts of transgenes that drive Alzheimer's disease pathology, Parkinson's disease pathology, driving cancer in these animals. And we're trying to humanize the immune system by either immunizing the mice or exposing them to pathogens to more mimic what goes on in a human lifestyle. So all of these are available and turning out to be quite interesting. I think this has been a huge gap in the field, that we don't have aging physiology affecting all of these age-related diseases. We're also doing tissue-specific deletion of ERCC1. So for example, if we knock out ERCC1 in the myocytes, we get heart failure, podocytes, we get kidney disease. Literally every time we touch these mice, we do get an age-related disease. We're also doing this in very cell type specific ways within the central nervous system and immune system. So in summary, very quickly, I believe strongly that endogenous DNA damage or genome instability plays a causal role, not just an ancillary role, in driving aging and most age-related diseases. Clearly, endogenous DNA damage is a driver of cellular senescence in vivo. Genome instability can initiate most, if not all, of the other hallmarks of aging. And I think this illustrates their interconnectedness and gives us a handle on therapeutic approaches. These mice offer, I think, a unique model of spontaneous accelerated aging and also comorbidities. That's what we're kind of lacking in our animal models, and that's what an old person looks like. So I think these are valid for testing virtually any class of geroprotectors. Okay, my last and provocative slide, I hope. When I looked at the clinical trials that are ongoing in the geroscience space to date, what's intriguing to me is all of them are clustered on these antagonistic or sort of responsive pathways to the primary defects. And so when you stop to think about it, what I came up with is I think the primary hallmarks, which are the ultimate instigators of aging, they're very, very hard to measure, right? You really need a specialized lab to measure DNA damage well or autophagy well. I think they're very tightly regulated. I think they're probably evolutionarily optimized because you need some DNA damage to evolve, but you don't need too much because you're going to get senescent and other things. I think they're very difficult to target and regulate pharmacologically. And I think another point to think about is overshooting your correction will have negative consequences that can be pretty severe. So anyway, happy to debate any of those with you. I'm extremely grateful to my lab, postdocs up top and bioinformaticians, my mouse team in the middle and the neuro group, the bottom row graduate students and undergraduates, which are just amazing, and I'm grateful to funding sources. Thank you very much for your attention.
Thanks, Laura, for a great talk. We have time for a question. Morten, and then...
Okay, great. Thank you so much, Laura. Really fantastic. I mean, I like that model very much. I was wondering about the endogenous DNA damage and how much you can attribute specifically to these oxidized lesions, or whether or not there could be other stuff that's also going on.
No, that's an incredibly important point. This is just the needle in the haystack, those cyclopyranatics. It turns out because of that extra bond between a base and a sugar, they're very stable, so they're the easiest ones to measure. It's impossible. There are hundreds and hundreds of lesions. And I've spent my entire lifetime trying to track down assays or people who could measure them, also trying to figure out are cross-links lethal and others not. So my advice to young people, don't go there. It's very expensive, very hard, and hard to do. All right, help me then. Thank you.
Don't tell people in my lab, thank you.
That was absolutely superb, Laura. Very quick question. What do you think the route is by which the mitochondria are so disrupted, given ERCC1's predominantly or overwhelmingly nuclear function? What do you think is going on there? What's the story?
Yeah, it's a great question. Obviously, signaling. There's no way it's mutations leading to disruption of electron transport chain. Clearly signaling. My personal belief is that all of the bad things that happen downstream of DNA damage is driven by the signaling pathways. We don't know them all, but that's p53 dependent, probably via ATM and simulation and signaling to the mitochondria. So that DNA damage response is a bad actor when it's on too long. Thank you.
Thanks again.
Thank you.
And